Good evening, everybody. Today is week 10 of the 12-week course, NOC 25 bt19 Enzyme Science and Technology, which is taken by Professor Vishal Trivedi of IIT Guwahati. I'm Sanket, PMRF Fellow from Kusuma School of Biological Sciences, IIT Delhi. Today's session is about enzyme inhibition kinetics. So we will be talking about enzyme inhibition kinetics today. And as usual, we will be discussing a couple of um, slides about the theory, and then we will go with the question and answers, the quiz, the model assignment. So we'll just wait for a couple of minutes. So enzyme inhibition is basically uh, done by an inhibitor, which either it's a chemical agent that inhibits or poisons the enzyme, basically slows down enzyme function. So we talk about the types. They can be of two types, either irreversible or it could be reversible, right? So if an inhibitor, let's say I, is irreversible, so it forms the complex enzyme inhibitor complex, and this enzyme inhibitor complex is a stable complex. Whereas if it is reversible, it forms an enzyme inhibitor complex which can be reversed, right? Um, Now, how to understand whether the inhibitor is a reversible inhibitor or an irreversible inhibitor? One of the ways is dialysis, where the inhibitor concentration is brought down and you see if the effect is reversed or not. By that, we can do it. Another one is gel filtration chromatography. In gel filtration chromatography, you have an axis for absorbance. Sorry. So absorbance and then the elution time. And you get peaks like this. So if let's say this is the enzyme peak, this is the inhibitor peak. If the enzyme is non-functional, then it is a irreversible inhibition. We'll see how that happens. If it is functional, then it is reversible inhibition. Another way is by performing dilutions. So by that, we can actually find out. So again, talking about the classification of enzyme kinetics, it could be irreversible or reversible. We already know that. So enzyme plus inhibitor can form different kinds of bonds. It could be electrostatic bonds. It could be hydrogen bond, stuff like that. And it can be either a competitive inhibition, especially the ones which are reversible, competitive inhibition, non-competitive inhibition, and of course, the third one is uncompetitive inhibition. Right? So when we are talking about irreversible inhibition, it's usually permanent, right? So it destroys a functional group necessary for catalytic activity. So the examples could be DFP or diisopropyl chlorophosphate. It inhibits acetylcholine esterase. This is important for nerve neurotransmission. Similarly, iodoacetate, it inhibits OH group of serine, SH group of cysteine, and imidazole group of histidine. DFP also inhibits serine proteases like trypsin, chymotrypsin, and cyanide. It inhibits cytochrome oxidase, which is involved in the electron transport chain, important for metabolism. Fluoride, it inhibits enolase. So it removes magnesium and manganese ions, which are important for the active site of the enzyme. So if we talk about neurotransmission, how does that work? So there is a neuron. So this is a neuron. And then there is a postsynaptic receptor. Fine. There is glutamate, acetylcholine, and other neurotransmitters involved. Now 5-HT6 receptor is involved for release of acetylcholine. It is blocked by RBT101. This inhibits this receptor. Fine. Similarly, choline esterase as we already know is inhibited by donespin. So it prevents breakdown of acetylcholine. Breakdown of acetylcholine. Now, if there is an increased concentration of acetylcholine, that leads to improved cognition. And it leads to improved cognition. Now, there's a special kind of inhibition called suicide inhibition. So what happens in suicide inhibition? It's a specialized form of irreversible inhibition. Fine. If we look at its properties, then it's a mechanism-based inhibition. Enzyme's own reaction mechanism inhibits it. So the inhibitor, that is a structural analog usually, is converted to a more effective inhibitor due to the action of enzyme. And essentially what happens is that the enzyme commits suicide. Right? And we will see some examples to understand how this happens. So if we look at some example, so xanthine oxidase is inhibited by allopurinol. So what happens? Allopurinol is converted due to xanthine oxidase into alloxanthine. And this is a more potent inhibitor of xanthine oxidase. It inhibits xanthine oxidase. Similarly, arachidonic acid to prostaglandin cyclooxygenase is inhibited by aspirin. It is commercially available. 5-fluorouracil. 5-fluorouracil is converted to 5-fluorodeoxyuridylate. This inhibits thymidylate synthase. Okay, thymidylate synthase, and it inhibits nucleotide synthesis and it's used in cancer. Right. So if we look at certain therapeutic Therapeutic uses of enzyme inhibition. Let's look at it. So we will see the clinical use, the enzyme inhibited, and the inhibitor. Fine. We will look at these. So the clinical use, for example, for epilepsy, we have GABA transaminase, and it inhibits. The inhibitor is GABA vinyl. Gamma vinyl GABA is the inhibitor. Fine. Similarly, as an antidepressant, the enzyme MAO is inhibited by tranylcypromine. The inhibitor is tranylcypromine and phenelzine. Similarly, antihypertensive, we use ACE as the enzyme. So captopril is the inhibitor. Similarly, for cardiac disorders, we have ATPase, which is inhibited by cardiac glycosides. For gout, xanthine oxidase, and we already know its inhibitor, allopurinol. Similarly, for ulcer, we have again ATPase, and it is inhibited by omeprazole. Right? So for a case study, which you can study well in your lectures about suicide inhibitor, by NAC, which is N-acetylcysteine. Now it is used as an adjuvant for treating malaria. Fine. So NAC is used as adjuvant therapy during malaria. So how it works is what we will see. So it's first of all, NAC is a byproduct of glutathione, GSH. It's a strong antioxidant. It protects cell from oxidative stress because it's a antioxidant. It reduces inflammation. Through reduced synthesis of TNF alpha, that is tumor necrosis factor. alpha. So what happens? Hemoglobin, it is converted to methylated hemoglobin due to RBC rupture. Okay, this happens due to RBC rupture during a malaria infection. Now, this leads to a high oxidative stress. Now, in this case, NAC comes in, and since it's a strong antioxidant, it's a reducing agent. So it leads to cell survival. So this is the basic schematic for the action of N-acetylcysteine. Now, talking about reversible inhibitors, so let's talk a little bit about reversible inhibitors. So among that, we'll talk about the first kind, that is competitive inhibition. Competitive inhibition. So what happens essentially in competitive inhibition is that there is an enzyme and the inhibitor competes with the substrate to bind to it. So if this is the enzyme, this could be substrate binding, or there could be inhibitor binding. So inhibitor, this is substrate. So if we look at certain examples of this, so what happens? How does this work? Let's have a look at it. So if we have enzyme plus substrate, it forms enzyme substrate complex, which leads to enzyme plus product, right? So there is a irreversible binding of enzyme plus inhibitor in this step. If we take at certain examples, we will see competitive inhibition of succinate dehydrogenase, by fumarate, malonate, and oxaloacetate. Fine, these are some examples. So this phenomenon can be reversed by increasing substrate concentration. Right? Competitive inhibitors are substrate analog. They resemble the structure of a substrate. So if we look at the kinetics of this, so enzyme plus substrate, we have already talked about it. So K1, K minus 1 forms enzyme substrate complex, which forms, if K2 is there, it forms enzyme plus product. And similarly, if we have inhibition here, which competes with the substrate, there is enzyme inhibitor complex formed due to the rate is Ki, enzyme inhibition complex, inhibitor complex. So if we want to find the rate of inhibitor concentration, it can be determined as EI, concentration of enzyme and inhibitor, and the complex altogether. We already know that V is equal to K2 into ES, and V max is equal to K2 into the total enzyme concentration, right? This is one equation, this is second equation. So we know that V by ET is equal to K2 ES by E. So the total ET is equal to E plus ES plus EI, right? We know this. So V by K2 into ET will give you S by Km into E by E plus S by Km into E plus I by Ki into E, or we can write V by V max is equal to S by Km 1 plus S by Km plus I by Ki. So I have taken out E from all the parts. So it is actually S substrate concentration by Km 1 plus I by Ki plus S. So if we treat this as alpha, we can write this equation, final equation, as V by V max is equal to S by alpha Km plus S. Fine. This is the inhibition constant. If we look at the graph, so of V versus S, so this is, let's say, V and this is S. If there is no inhibition, we will see a graph like this. And this is, let's say, V max. If there is inhibition, I represent it using red, then the graph looks like this. So the Vmax is remaining the same. However, if we look at the half Vmax value, which is Km, so that changes. So if this is Km and this is Km dash. So what we usually see is that Vmax remains unchanged, whereas Km increases in case of competitive inhibition, right? This is in case of competitive inhibition. If we see a Lineweaver plot, which is exactly the plot which we use to find out the type of inhibition, it is plot between 1 by V and 1 by S. We see in absence of inhibitor, this is, let's say, the plot, there is no inhibitor. And alpha is equal to 1, right? There is no inhibitor. This point is 1 by V max, and this point is minus 1 by Km, right? We know that already. If there is inhibition, so in presence of an inhibitor, so alpha could be, let's say, 4 here. So that's in the presence of inhibition, the Km value will obviously increase, whereas Vmax remains the same. So this is a Lineweaver plot for competitive inhibition. Now talking about the therapeutic uses, we have the drug, the enzyme inhibited, and the clinical use. If we look at these 3 points, 6-mercaptopurine, 5-fluorouracil, azathioprine, uh, cystonic arabinoside, then we have all these, they inhibit different enzymes like adenyl succinate, synthetase, thymidylate synthesis, phosphoribosyl aminotransferase, and DNA polymerase. All are used in case of cancer. Similarly, there is acyclovir, which also inhibits DNA polymerase used for as an antiviral agent. Similarly, there are multiple drugs for this, right? Now, talking about non-competitive inhibition, it is reversible but cannot be reversed by substrate. The inhibitor binds at site other than the active site. Both enzyme and enzyme substrate complex. So an example for non-competitive inhibition would be allosteric inhibitors, uh, very common. Fine. They alter the conformation of enzyme. They're usually naturally occurring metabolic intermediates. Okay. So if you want to see how the kinetics works, so enzyme plus substrate can form the enzyme substrate complex, which forms the enzyme plus product. And these are the, so here also there could be inhibition, like competitive inhibition, Ki. And here also there could be inhibition, which is non-competitive. Let's say Ki dash, right? So we know using Michaelis-Menten kinetics that Ki is equal to enzyme by Ei, whereas Ki dash is enzyme right? So we can write V by Et is equal to K2Es by this is the total enzyme, or V by the max is equal to the substrate by Km. If we mark this as alpha and this as alpha dash, so Vmax can be written as V by Vmax. The final equation is substrate concentration by alpha Km plus alpha dash S. if you look at the graph of this kinetics, so if this is V and this is S, inhibition looks like this. Usually, this is half, this is V max, and this is, let's say, V max. So Km doesn't change. So in this case, Km remains unchanged, but V max decreases. Fine. So if we see a Lineweaver-Burk plot for this, will look like this. This is in absence of an inhibitor, and in the presence of an inhibitor, look like this. So this is 1 by V max. This is 1 by V max dash, right? And the Km values remains unchanged. So there are some examples of this. So if we get a comparison, okay, we do a comparison by the criteria, and then there is either competitive or non-competitive, how will it look like? Let's see. So if we divide it based on the active side, it is usually on the active side, and this is on sites other than active site. The structure, if we look at, they are substrate analogs in competitive inhibitors, and in non-competitive, they are unrelated structures. Fine. Inhibition type, if we look at, they are reversible, and here they are usually irreversible. If we have excess of substrate, then the inhibition is relieved. In case of competitive, there is no effect in non-competitive. Km increases in competitive inhibition, it is unchanged in non-competitive inhibition. Vmax is unchanged in competitive inhibition, it decreases is in non-competitive inhibition. Then there is something called uncompetitive inhibition. So what happens? Enzyme only binds to the enzyme substrate complex. So enzyme plus substrate, it gives you enzyme substrate complex, it gives you enzyme plus product, and the inhibitor only binds here, right? And this leads to no product formation. Fine. And the final equation looks like very similar to competitive inhibition. V by V max is equal to S by alpha Km plus S. So if you look at the Lineweaver-Burk plot for this, so it will look something like this. 1 by V, 1 by S. If there is no inhibition, this is no inhibition. If there are inhibitors present, they will be parallel lines. The slope is Km by Emax, right? Now let's solve the questions if the substrate concentration is much higher than the Michaelis-Menten constant, what is the relation between the reaction rate and maximum velocity? So in that case, the reaction rate and maximum velocity are equal. Now, the maximum velocity of an enzyme catalyzed reaction is 50 micromole per minute. The Michaelis-Menten constant is 10 millimolar. Calculate the initial reaction rate when substrate concentration is 20 millimolar. So to find out, we have what have been given to us. Vmax is 50 micromole per minute. Km is 10 millimolar. And S is given as 20 millimolar. So if we calculate this, so V is equal to Vmax into S by Km plus S. This is Michaelis-Menten equation. We can just put the values here. It's millimolar, so we'll convert it into micromolar. Okay, and then we have both in millimolars, Km and S. So I will write it as 10 plus 20. So 10 to the power 3, 10 to the power 3 cancels out. We have so it would be 33.33. So this is the answer. Micromole per minute. Catalytic efficiency Kcat by Km is given as 1000. We have Kcat value is also given as 500. So what we need to find out is Km. That would be Kcat by Kcat. Sorry. So 1000 by 500 will actually be 1 by Km. So 1 by 2, it would be 0.5 millimolar. The turnover number of an enzyme is defined as the maximum reaction rate, ratio of Vmax to Km, number of substrate molecules converted to product per enzyme active site per unit time, or the rate constant. So it is the number of substrate molecules converted to product per enzyme active site per unit time. The type of enzyme inhibition can be overcome by increasing substrate concentration. That happens in case of competitive inhibition. The purpose of using a double reciprocal plot, also known as the Lineweaver-Burke plot, is to identify the type of inhibition, right? So Vmax is given. Okay, in this case, we have to use the formula the Michaelis-Menten equation by Km plus S. And using this, if you calculate the initial velocity of the reaction, with 36 micromole per minute. The Lineweaver-Burke plot for an enzyme catalyzed reaction, the x-intercept is -2 millimolar and the y-intercept is given. Calculate Km. So if this is the Lineweaver-Burke plot, using the x-intercept is usually the Km value. It's already given, minus it's usually in the negative axis. So the Km would be 2 millimolars, right? That's the answer. Question 9, in reversible non-competitive inhibition, the inhibitor bears structural resemblance to the substrate? No. Inhibitor lowers the maximum velocity attainable. Yes, that is true. Km is decreased, Km is increased. No, Km, there is no change in Km. The enzyme follows Michaelis-Menten kinetics. The reaction velocity at substrate concentration Km by 3 is what? So if we use the formula, V is equal to Vmax into S by Km plus S. So we have been given that substrate concentrations Km by 3. So let's do that. Vmax into Km by 3 by Km plus Km by 3. So if we do it, we solve this. By 3 gets cancelled out, Km gets cancelled out. V is equal to Vmax by 4. So that is the answer. In order to catalyze a reaction, an enzyme is required to be saturated with substrate, decrease the activation energy, increase the activation energy, or increase the equilibrium constant. It actually decreases the activation energy. It's very similar to this graph. So if in absence of an enzyme, substrate forms a product with a higher activation energy. In the presence of enzyme, it will lower the activation energy. That is the idea. So this is the activation energy. This zymogen is the active form in enzyme, complex form between enzyme and substrate, inactivated form enzyme, activated by cleavage, or unfolded form in enzyme. Zymogen is an inactivated form of enzyme which is activated by cleavage. Lower the Michaelis-Menten constant shows greater affinity of the enzyme for substrate, less affinity for enzyme for the substrate. Enzyme is allosteric, enzyme is non-allosteric. So it tells us whether it has a greater affinity of the enzyme towards the substrate. That is the main idea. Because we know that, if you see the graph between velocity and substrate concentration, the Km value actually tells us how much affinity the enzyme has towards the substrate. If the Km value is, if an enzyme has a lower Km value, that means, so in this case, it has a lower Km value, it means it has higher affinity towards the substrate. The greatest increase in reaction rate will be by increasing the substrate concentration by a factor of 1.5, increasing the substrate concentration by a factor of 2, increasing the Michaelis-Menten constant by a factor of 2, or increasing the maximum velocity by a factor of 2. So for this, we need to have the Michaelis-Menten equation. So let's write that. So V is equal to V max into S by Km plus S. So if we increase the substrate concentration, that will increase the numerator and denominator both. So see, that will be a complex situation. So the reaction rate will increase, but there are that depends. Now coming to the Michaelis-Menten constant, if we increase this, the reaction rate will decrease rather. But if we increase the maximum velocity, then that will definitely increase the reaction rate. So D would be the right answer. The ratio V by V max when S is 5 km. So we have to calculate this. Let's do that. So V is equal to V max into S by km plus S. We know this is the Michaelis-Menten equation. Now V by V max is the ratio that we need to find out, and the substrate concentration is given as 5 km. So let's do that. So that would be S. Let's put the value of 5 km. That would be 5 km by km plus 5 km. So this would be 5 by 6. The answer would be 5 by 6. Km will cancel out. So 5 by 6 is the answer to this question. Okay. With this, we are done with today's quiz as well. So if there are any further questions, you can ask regarding this week. Is there any question? Anything from the slides, anything that you might have not understood?
Sir, question number 13.
13. Yes. So the Km value in a Michaelis-Menten equation, V is equal to V max into S by Km plus S. This Km value actually tells us the affinity of enzyme towards substrate. So if this value is lower, then the enzyme affinity will be higher because the reaction rate will increase because it's in the denominator. Is it clear?
Yes, sir.
Okay. Any more questions?
No, sir.
Okay. Okay. Okay, then if there are no further questions, I would be ending today's session, if that is okay with you.
Yes, sir.
Okay, thank you so much for joining.
Thank you, sir.
Bye bye. We'll meet next week, same time.
Okay, sir.